Welcome to our fourth podcast on the digestive system, and today we'll be covering the salivary glands and the pancreas. These associated glands are critical during the process of development, beginning the chemical process, and then the pancreas adding to those chemicals after we've come through the acidic environment of the stomach. So with these objectives, what we're going to accomplish is identify the parenchyma and the ductal system of the salivary glands. That will histologically allow us to identify them as the different glands we have: parotid, submandibular, sublingual. We'll also compare and contrast the three salivary glands. So if they're mucus versus serous products, or the type of ducts that are present, will help us do just that. We're then going to look at the pancreas. There are a number of cells in the pancreas, and not the least of which we are going to compare and contrast the exocrine portion of the pancreas from the endocrine cells that we have in our islets of Langerhans. So as we begin to look at these extramural glands in the digestive system, we're going to be looking at the salivary glands, pancreas, and then our fifth podcast that we'll save for later, we're going to be looking at the liver and the biliary system. So with our major salivary glands, if you recall, in the lip and the tongue, we saw a number of minor salivary glands. But what we're going to see here is we're going to have some sort of combination of serous and mucus cells, and the percent of each will help us identify the respective glands. Now, we can also have some, and we're not really going to identify them, but some are referred to as seromucus because they can both have a mucal component and a protein component. Most of the time when we talk about mixed glands, we're talking about those mucus acini with the serous demilunes. Now, we're also going to mention here the myoepithelial cells. These will be peripheral to the parenchyma, and they do have contractile properties that assist in the expression of the secretory product into the ductal system. Now, with the ducts, we are going to have sort of a hierarchy of our designation histologically. The beginning of the ductal system is going to start with an intercalated duct. Now, these are going to be the first parts that actually insert themselves into the lumen of the acinus itself. So the intercalated part means it's within the acinus. As this leaves the acinus and moves out, it's going to widen its diameter, and it's going to become a striated duct. Now, the reason we call these striated is as we look at the cells and the basal surface of these cells, they're going to increase their surface area by folding themselves a number of times. And at the light level, this is going to appear as a striped or a striated pattern. Now, it's within this basal compartment that we are going to have these sodium-potassium pumps, and we are going to pump this material into the connective tissue. Now, that offsetting that we're going to have with the sodium potassium as we're pumping the sodium into the connective tissue, that's going to allow us then to pump the potassium and the bicarb into the lumen of our ductal system as we express these materials. So here we see a diagram of the ductal system. We have a couple illustrated here. We have a serous acinus, which is going to be darker in color. Here we have a mucus acinus, and I wish they had made these a different color, the serous demilunes out here. Here's the lumen of our acinus, and we can see these ducts inserted within and the first portion of the ductal system. These are the intercalated ducts. And so a number of these join together into the larger striated duct. Now, these are just examples of serous cells, the ductal cell of the intercalated duct, the mucus cell, and the striated duct, which I wish they had a better illustration showing the folds. They attempt to do that. You can't quite see them as striped as I had hoped they would see. But here in our ductal system, we can recognize the parenchyma. These are going to be the serous parenchyma within our gland. These very round structures with a round luminal center, these are going to be the ductal system, but then you can make out the striations that we have in these larger ducts, and these are in fact the striated ducts cut in cross section within this gland. Now again, as we move to ever larger and larger diameter ducts, we're going to move from a striated to an intralobular, that become larger interlobular, then intralobar, interlobe, and then finally we get into our terminal duct. And this is going to be the duct that empties into the particular target area where material is delivered. In the case of our salivary glands, that's going to be into our oral cavity. Now, we're not going to identify these in histological sections. We're really only going to see intercalateds and the striated ducts. Now, saliva certainly is going to be present for the beginning of chemical digestion, but it's also present for lubrication. We're also going to have this fluid washing through and cleansing our oral cavity, our tongue, and our teeth. But then we have a number of materials that are going to help in antibacterial activity. As the material stays around, it's going to be a nice environment to grow bacteria. So, in our saliva, we have materials such as lactoferrin, which is going to sequester iron and prevent it from being used by bacteria for their metabolism. Lysozyme, which is going to degrade bacterial capsules. We have thiocyanate ions. Now these, it's going to almost like be a cyanide pill. These are going to penetrate the bacterial cell wall and the membrane of the bacteria, and therefore cause the bacteria to die. Now we're also going, as I mentioned, begin to dissolve the food into this liquid material, into the bolus. We are going to begin this chemical digestion process. A number of enzymes, certainly not the least of which is salivary lipase, which begins to break down lipid. But we also have a salivary amylase, which is going to begin to break down carbohydrates, and this is talin. Now, the fluid environment in creating this bolus of material, that's going to aid us in swallowing, the lubrication, the breaking down of the food, that assists. But also we're going to have materials present in our saliva such as clotting factors, epidermal growth factor. These are going to assist in the process of clotting, because with all the frictional activities in the mouth, and you've seen your gums bleed even when you brush your teeth, we're going to have bleeding in the gums periodically. So this is going to assist in fixing any vascular injury we might have in the oral cavity. Now, saliva as it's produced is referred to as primary saliva. And initially this material is fairly isotonic with what we see in the plasma. But as you can see in the illustration, as the material is expressed into the ductal cells have the capability to alter this material. And so as you can see, sodium is pumped out of the saliva. Potassium and bicarb are injected into the saliva, and you can see chloride as well is shifted more out of the saliva and into the tissues following sodium, as it typically does in many organs such as the kidney. So now let's turn our attention to the histology of these glands themselves, and the first we're going to cover is the parotid gland. This is the largest of our salivary glands. However, it does not produce the majority of saliva. Only 30% of the saliva is produced by the parotid. Now, it is positioned on the side of the face, just near the jawline, and it's positioned over the masseter muscle, which is the largest muscle present in our jaw. Now, the easiest way to identify the parotid when we determine we are looking at a gland and lots of acini and ducts is the fact that the parotid gland consists of purely serous acini. So as we look at a section of parotid gland, it's going to be fairly easy to pick out that material. Now, in the parotid gland, we have very long intralobular ducts. We're going to see in other glands we have longer striated ducts, so we'll see them more. So whatever you see in section, that also is going to assist in your identification of the organ itself. Now, since these are in fact serous, we know that they are zymogenic, producing enzymes, and typically these cells are producing the enzyme talin. This is going to be that salivary carboxylase. It's breaking down the carbohydrate material. Now, as we continue along the pathway, we will eventually end in our terminal duct, and this is going to be in Stenson's duct, and this is going to open into our oral cavity on the inside of the cheek, opposite the second molar. So in our illustration here, we can see on both sides, we can see the parotid gland. Here, that's number 1. And here we can see the parotid gland. And this is the one that becomes enlarged whenever children get the mumps. And from this, here is Stenson's duct projecting into the oral cavity, again, opposite that second molar, I'm not sure if that's going to be illustrating Stenson's duct there or not, but you can see that injury. Now, as we look at a section, much of what we're looking at as far as this lighter stained tissue are going to be blood vessels or our ductal system. And we can see filled with blood. We can see some arteries here, possibly a vein in many of these. It's pooling of the blood. We have septa, connective tissue septa running through here. But as you look at the glandular parenchyma, notice that it is this darker color, other than the lighter color, which are going to be some of these ductal portions. But all of that is darker. That is serous secretion, and this is a 100% parotid gland, serous secreting. And on a practical, you're going to see much higher magnifications of this, and we'll look at that in lab as well. Now, the second gland we're going to cover is the submandibular gland. Of course, with the name, we know where its location is, beneath the mandible. And this one is not the largest, however, it produces the bulk of the saliva, over 60%. Now, the majority of our acini here are going to be serous, but we will have mucus acini, maybe upwards of 10%. But the other characteristic that we're looking for in these sections, we're going to see a number of striated ducts. They have very long striated ducts. A lot of modification occurs here, but since they're so very long, we're going to be more apt to see them in cross sections, longitudinal sections. We didn't see many, if any, in the parotid gland because they are short to absent in the parotid gland. Now, the terminal duct here is called Wharton's duct, and it opens at what is referred to as the sublingual caruncle. Now, if you think of the frenulum of your tongue, which is going to be this little strip of tissue, you're probably familiar with it, that connects the ventral surface of the tongue to the floor of the oral cavity. This is the frenulum of the tongue. And here at the base, you're going to see this enlargement, and that's the caruncle. And you're going to see these openings. These are the openings of Wharton's duct in the sublingual caruncle. And one way that you can probably have noticed this before, those people that can express material from their mouth by sort of squeezing their tongue, and I believe that it's colloquially called gleeking, that is the expression of salivary material from Wharton's duct, and that's where it's coming from. Now, this is just an example to remind you of what mucus acini look like. And so here we have mucus acini, some serous demilunes, and then here we have some just pure serous acini. And in and this is going to be a duct, striated duct. We can see the striations here along the base, and this is a longitudinal section through those. And if we're looking at submandibular gland, you're going to see lower magnification than this, so you can appreciate the percent of the acini that are serous versus that is mucus. And remember, 10% will be mucus for the submandibular gland. Now, the last salivary gland we will cover is the sublingual gland. And in this is the smallest of the three, produces very little, if anyway, 5% of the total mucus. And in this case, most of the sublingual is going to be mucus acinus, and we're going to have a third of that as serous demilunes. Now, there are a number of ducts that are entering into the oral cavity from the sublingual, but the largest one is called the duct of Bartholone. And it is going to join the submandibular duct and drain through the sublingual caruncle as well. And so here is a sample of a primarily mucus-looking region. As we can see, all of the white mucus acini. This looks almost 100% mucus. You can see some serous material here, but certainly we're going to be looking for the serous demilunes, and you can see this darker material on the edge, and that's primarily what we're looking for when we're identifying sublingual gland. Now, the other structure that we really think of as a gland when we're talking about the digestive system is the pancreas. It's going to produce much of the same material that's present in the saliva. However, think about what's happening to this material as it moves through our alimentary canal. We first inject the saliva, a lot of enzymes, which are protein. the first thing they're going to encounter is the stomach, the acidic environment of the stomach. And so many of these enzymes are going to be neutralized in a very short period of time. So the pancreas is going to inject another round of enzymes after this material has been hydrolyzed in the stomach by the hydrochloric acid, after that acid has been neutralized by the bicarb produced by many of the cells in the pyloric region of the stomach in the beginning of the duodenum. So in the pancreas, we are a compound tubuloacinar gland producing on the order of 1.2 liters of fluid material per day, and that's typically referred to as pancreatic juice. And this juice is going to be injected into the alimentary canal at the level of the duodenum. Now, our acini are going to be fairly large. 40 to 50 cells are going to make up this acinus in the pancreas. And we're actually going to have cells that define the lumen of this acinus, and these are called centroacinar cells. They're going to line the lumen. They are going to define the lumen and the beginning of the ductal system, and this is the endocrine portion of the pancreas. And when you see centroacinar cells in the middle of an acinus, rather than just an open space, that alone will tip you off you're looking at the pancreas. Now, these centroacinar cells are going to respond to many hormones. They can even respond to neurotransmitters. So we're responding to secretin, which is going to be a hormone, and we're responding to acetylcholine, which, remember, is parasympathetic rest or digest, that causes the expression of this pancreatic juice, this enzymatic-rich material, into the alimentary canal. Here we can see the pancreas is situated just in the bend of the duodenum. We have the main pancreatic duct running through the body of the pancreas. Here's the common bile duct coming down. Once again, as we look at the pancreas itself, here's our intralobular duct that is taking material away. Here we have our acini. This is going to be the exocrine portion in blue that we see. Lining inside, you can see these yellow cells. These are the centroacinar cells that are going to define the lumen and be the beginning of the ductal system as we transport this material out. Now, it's bluish in nature, but for all intents and purposes, since these are enzymatic secretions, this is going to look like a serous gland, a pure serous gland. However, because of the exocrine portion of the pancreas, the pancreatic islets, or the islets of Langerhans, these are going to also be extremely characteristic of helping us define the organ as a whole. So here we're looking at a purely serous acinus. Very easy to see that. You can see all these zymogenic granules that are present here. And then all of a sudden, when you're looking at an acinus, you see the nucleus of this cell right in the middle, and here we can see the beginning of the ductal system. Here we have another. Here we have another. These are the centroacinar cells that are present in the middle. Now, in addition to all of the enzymatic material that's present in these cells, the acinar cells, or the parenchyma of the gland, they're going to have receptors for cholecystokinin. Cholecystokinin, remember, is going to cause the spasmodic contractions in the gallbladder, and we'll talk about that in a bit. And that's also going to express the material to the duodenum. So when cholecystokinin is produced by the stomach, it not only is going to affect the gallbladder, but it will also affect the pancreas. Now, we will have visible striated ducts, but again, these are going to have very long intercalated ducts. And these intercalated ducts in the form of the centroacinar cells, again, are the main identifying characteristic for pancreatic tissue. Now, the terminal duct, as we saw, is the main pancreatic duct. It's going to join with the common bile duct, and where it enters in the duodenum is a very complex structure that's called the ampulla of Vater. So here we see a little bit more detailed illustration of what we're talking about. Here we can see the acini that we have, our centroacinar cells in the beginning of our ductal system as they're draining out. This will flow into the main pancreatic duct. The main pancreatic duct comes down, and here we have this ampulla of Vater. This ampulla of Vater is going to be a common sort of receiving area before we empty material into the duodenum. And on the inside of the duodenum, we can see the papilla of Vater. But this ampulla of Vater, this is a very open area, and we can see bile coming down the common bile duct. We have material coming from the pancreas, and it's going to be muscular sphincters within this ampulla of Vater and prior to the papilla of Vater that are going to help us regulate the passage of material in and out of the duodenum. We'll talk more about that when we get to the system on liver and the biliary apparatus. So as we look at a low magnification view of an organ, we're first struck by how dark this material is. And so as we begin to break this down, we're looking for distinguishing and identifying characteristics. And all of a sudden we see these fairly large, lighter colored round regions. This is going to be the first indication that you're looking at pancreas, because these are in fact the islets of Langerhans. Now, in a different field, we can recognize all of these serous acini, and even though we can't really make out the central acinar cells, the islets of Langerhans, the pancreatic islets, are clearly visible. And so this should be a cakewalk for you to identify this as pancreas. Once again, looking at a purely serous type of gland. Here we see our serous acini all around, but now in the middle of the lumen we see these nuclei, and those are going to be our centroacinar cells that are present, letting you know that we're looking at pancreas. Now, a lot of material that's produced that makes up this pancreatic juice. These parenchymal cells are going to produce pancreatic amylase. It's like taline in the saliva, but this is pancreatic enzymes that break down carbohydrates. We have pancreatic lipase. Then we have RNase, DNase, trypsinogen, and chymotrypsinogen, which are inactivated proteases. They're going to be activated as this material is produced and expressed into the duodenum. And then we also have procarboxypeptidase. Now, what this is going to do is this is going to hydrolyze that first peptide of the amide bond at the carboxyl end, which is really almost a cap. And this is going to really prevent degradation of a lot of proteins. And so this procarboxypeptidase basically unlocks the ability of other enzymes to come in and help break down these proteins in a much more efficient way. And this procarboxypeptidase, as you can see here, it's got a higher preference for aromatic and branched hydrocarbon chains, which are more difficult to break down those amino acids than we have with purely unbranched hydrocarbons or more simple structures to the R groups of many of these amino acids. Now, also within the pancreatic juice, we have elastases, and then we're going to have trypsin inhibitor, which essentially is going to be present to slow down the reaction so that the trypsin doesn't auto-digest itself before it completes the project of digesting the material that's been eaten. Now, the centroacinar cells and the cells of the intercalated duct, they also are going to assist in the modification of this pancreatic juice. They are enzymatic, so they're serous, but it's also bicarb rich. It's important to remember that we're emptying these materials into the duodenum, which just received this chyme from the stomach, and there may be hydrochloric acid remains. So it's important to have this bicarbonate-rich material to neutralize any acid that remains so that we don't damage or inactivate the enzymes before they can perform their function.